everyone. My name is Beth Moore, and I'm the chair of microbiology and immunology at the University of Michigan. And I'm also a member of the review panel for the Pulmonary Fibrosis Foundation, and I got the pleasure of reviewing the many abstracts that were submitted for the meeting. Today, I have the pleasure of introducing you to our 5 poster award winners, and I'm going to just tell you their names, where they're from, and the titles of their posters before we get into their presentations. So our first place winner this year is Jason Gokey, who is a research instructor at Vanderbilt University, and the title of his poster is "YAP regulates alveolar epithelial cell differentiation and AGER via NFIB, KLF5, and NKX 2.1. Try to say that 3 times fast. It's a great poster. I hope you'll join us for it. Our second place winner was Luis Rodriguez, who is a postdoctoral fellow at the Perelman School of Medicine at the University of Pennsylvania. The title of Luis's poster is "Leveraging the Surfactant Protein C I73T mutant surfactant mouse model for preclinical intervention studies to benchmark idiopathic pulmonary fibrosis therapeutics and looking at the effect size of nintedanib and TGF-beta pathway inhibitors." Our third place winner is Yi Yao, who is a research scientist and instructor at the Henry Ford Health System, and she will be presenting her poster on the loss of microRNAs in alveolar macrophages attenuating bleomycin-induced pulmonary fibrosis. Our fourth place, or our honorable mention, I guess we're calling it, is Margaret Thomas Freeberg, who is a postdoctoral fellow at Patricia Symes Lab at Virginia Commonwealth University. Her poster is entitled "Mechanosensing by Piezo2 is associated with metabolic reprogramming in human lung fibroblasts." And as I understand it, Dr. Freeberg is a little bit indisposed, possibly having a baby today. Congratulations to her. And so I believe we will hear from her mentor, Dr. Syme, in her place. And our second honorable mention goes to Dr. Bhavika Kaul, who is an assistant professor at the University of California at San Francisco, who will present a poster title called "Agent Orange Exposure is Associated with Subsequent Development of Idiopathic Pulmonary Fibrosis Among United States Veterans." So I hope you will stay on and listen to the presentations of these fabulous posters. We'll be back at the end of those to have a question and answer session. And so please be ready with your questions and comments. Enjoy.
Hello, today I would like to share with you our recent work understanding the role of the hippo-YAP signaling during alveolar epithelial cell differentiation during development and injury repair. The alveolar epithelium or the alveoli is the functional gas exchange unit in the lung, and it consists primarily of 2 alveolar epithelial cell types. We have the alveolar type 1 cell here, labeled in gray, that conducts gas exchange in close proximity to the alveolar capillary, here seen in red. And then there's the alveolar type 2 cell, here in yellow, that functions to make and secrete pulmonary surfactants and lipids to maintain alveolar homeostasis, and also acts as a facultative progenitor cell during injury to regenerate both the type 2 cell population and the type 1 cell. Now, the lung demonstrates a remarkable capacity to regenerate following injury, as the lung is readily exposed to environmental toxins and such as it is as you breathe in the air. This includes things like cigarette smoke, seasonal viruses such as influenza, as well as the current COVID-19. During the alveolar injury and repair, the alveolar type 2 cell serves, as I said, as a facultative progenitor, in which it's able to repopulate both alveolar epithelial cell types. In the airway, the basal cell is the classical alveolar, or the classical progenitor cell, where it's able to repopulate both ciliated and secretory cells. This failure of alveolar epithelial repair is believed to be a driving cause in interstitial lung diseases, including idiopathic pulmonary fibrosis. Here in the normal healthy donor lung, you can see labeled in green, second harmonic resonance showing collagen deposition and the structure of a normal healthy alveoli. And in red, we have ABCA3 positive type 2 cells. However, in idiopathic pulmonary fibrosis, or IPF, you can see the loss of this alveolar structure. And in white, you can see the deposition of smooth muscle actin, indicating fibrotic scarring. IPF affects roughly 100,000 patients in the US and Europe, and it causes the progressive decline of respiratory function and is usually fatal within 3 to 5 years of diagnosis. Hallmarks of IPF include loss of alveolar epithelial cell identity. In the normal healthy lung, we have both the presence of type 1 ABCA3 positive cells here in green, as well as HOPX positive type 1 cells that conduct the gas exchange labeled in red. There's also a proximal distal pattern as identified here with HOPX positive type 1 cells being not proximally located or not near P63 positive basal cells, which are present in the proximal epithelium. In contrast, in idiopathic pulmonary fibrosis, we can readily detect both HOPX and ABCA3 positive cells, indicating abnormal epithelial cell differentiation as they express markers consistent with both cell types. We also have a loss of this normal proximal distal patterning with P63 positive proximal epithelial cells in close proximity to our red HOPX positive type 1 cells. We performed single-cell RNA sequencing on isolated type 2 cells from normal and IPF lungs. And in our control normal type 2 cells, these cells express markers such as surfactant protein B and C, as well as ABCA3, consistent with a normal type 2 cell. In contrast, in the IPF lung, we found 3 distinct clusters of cell types, including a basal-like type 2 cell that expresses markers such as P63 and keratin 5, normally reserved for the proximal airway, as well as a goblet or secretory-like cell type in the distal lung that also expresses things such as secretoglobin, SEGB1A1, and MUC5B. There was also indeterminate cell types that express markers across multiple cell lineages. For example, they may express things such as surfactant protein C, as well as keratin 5 and secretoglobin SEGB1A1. To understand what's driving this abnormal epithelial cell differentiation, we performed pathway analysis, which predicted that the hippo-YAP signaling pathway may be driving this aberrant epithelial cell differentiation. Now, the hippo-YAP pathway is an ever-expanding pathway with increasing degrees of complicated signaling networks. However, in the most simplest form, there's the canonical serine threonine kinase, SDK3 and 4, also known as MST1,2 in humans, that phosphorylates LATS1,2, which then phosphorylates YAP. In the presence of this phosphorylation cascade, YAP is sequestered into the cytoplasm where it fails to interact and drive transcription. However, in the absence of this phosphorylation, YAP shuttles to the nucleus where it interacts with its binding partner TEADs to drive transcription. In IPF, or in a healthy donor lung, ABCA3 positive type 2 cells readily express MST1,2, again, the homologue of SDK3/4. And this is indicative of the brakes essentially being on the YAP pathway. Consistent with this, ABCA3 positive cells here in red, as labeled by immunofluorescence, have rarely detectable nuclear green YAP signals, suggesting again that the YAP pathway is not on. In contrast to this, in the IPF lung, ABCA3 positive cells seem to be lacking the MST1,2 expression, suggesting that the brakes are now off the YAP pathway. And this is consistent with the concept here that we have red ABCA3 positive cells, readily detectable green nuclear YAP staining consistent with an active YAP signaling. We then looked at YAP transcriptional targets, including AXL tyrosine kinase, which here you can see is pretty well low detected in the donor lung. However, in IPF, there's marked increased expression of AXL tyrosine kinase as identified by these white puncta through an RNAscope. Likewise, another YAP target, Ajuba, is rarely detected in the normal donor lung, whereas in IPF, again, in our red ABCA3 positive type 2 cells, we readily detect these white Ajuba positive nuclei, again, indicating that there's active YAP in the IPF epithelium. To determine the role of YAP in alveolar epithelial cell differentiation, we developed 2 separate models, one in which we're able to activate YAP specifically in type 2 cells using the surfactant protein C driver and knocking out SDK3/4, again, the inhibitors of YAP, to thereby activate the YAP signaling pathway in type 2 cells. And we also generate a YAP deleted version of this mouse where we can delete YAP specifically in type 2 cells. We then used IP injection of tamoxifen at postnatal day 3 to activate the YAP pathway, and we assessed the lungs postnatally and found that we are in fact able to induce, as seen here in these yellow cells, nuclear YAP localization in the YAP-active mouse, suggesting our model is working. Analysis of YAP-active and deleted mice shows that, consistent with recent reports, YAP activation induces increased number of type 1 cells. However, we also readily detect the presence of a HOPX positive here in green and surfactant protein C positive in red, both AT1, AT2-like cell. This is also demonstrated here with the other AG type 1 cell marker AGER with surfactant protein B. And this presence of dual positive type 1, type 2 cells is indicative of an incomplete alveolar differentiation. In contrast, YAP deletion essentially had fewer type 1 and fewer type 2 cells. We then wanted to assess this in vitro and generated alveolar epithelial cell organoids by isolating type 2 cells from either wild type, YAP active, or YAP deleted mice. YAP active type 2 cells preferentially differentiated into alveolar type 1 cells. In contrast, YAP deleted type 2 cells formed almost exclusively type 2 cells, failing to generate type 1 cells. To determine how this might be happening, we performed RNA sequencing on isolated YAP deleted type 2 cells and found that these cells have increased expression of mature type 2 cell markers, and this includes surfactant protein B and ABCA3. This existence of a enhanced type 2-like phenotype, or in words of a recent publication, exaggerated like type 2 phenotype, has been recently demonstrated by other groups. In contrast, YAP activation, RNA sequencing of YAP active type 2 cells showed the presence of type 2 cells expressing aquaporin 5 and AGER, canonical type 1 cell markers, as well as increased expression of secretoglobin cells, secretory cells, SCGB3A1, 3A2, and 1A1, as well as MUC5B, indicating that these YAP active type 2 cells have an indeterminate-like differentiative state expressing multiple lineage markers. To try to determine how this YAP activation state causes this activation of abnormal markers, we performed ATAC sequencing, or analysis of transposase accessible chromatin, to analyze the openness of the chromatin, and found that YAP activation opened approximately 3,000 promoters of different genes, and that these genes map back to cell cycle regulation, consistent with a well-known role of YAP regulating cell proliferation, and also map back to genes associated with AT2 and AT1 differentiation and development. Further analysis of these YAP opened chromatin regions showed that these open promoters were enriched for binding sites of transcription factors KLF5, the well-known binding partner of YAP TEADs, as well as NKX2.1 and NF1. To determine how these may be interacting, we co-expressed YAP, NFIB, KLF5, and NKX2.1 in the airway epithelial cell line HBEC3s, and found that by expressing these transcription factors, we're able to significantly increase expression of the type 1 cell marker AGER. Interestingly, using siRNA to inhibit TEADs, the YAP binding partner, we're able to significantly reduce that induction of AGER. To determine the functional role of this, we turned to the AGER promoter, in which we acquired a luciferase assay construct to determine functional binding to the promoter, and we found that when we co-express NKX2.1, KLF5, YAP, and NFIB, we're able to significantly enhance the promoter activity of AGER. We then identified binding sites for NFIB, NKX2.1, TEAD, and KLF5 in the AGER promoter region, and we used site mutagenesis to alter the NFIB, NKX2.1, or the TEAD binding site. We did not alter KLF5 binding site due to the numerous binding sites identified. We found that whenever we inhibit any one of these 3 transcription factor binding sites, we're able to significantly reduce the YAP-induced activation of AGER, suggesting our model here in which in some way YAP, TEAD, NFIB, KLF5, and NKX2.1 all functionally interact at the promoter region of AGER to activate transcription. So in conclusion of this work, we found that YAP activation causes widespread accessibility and openness of chromatin that was enriched for binding sites for the YAP binding partner TEAD, NFIB, KLF5, and NKX2.1. This led to increased number of type 1 cells, as identified by AGER and HOPX expression, as well as the presence of indeterminate type 2 cells expressing markers such as surfactant protein C, AGER, and SCGB1A1. And the YAP deletion leads to an enhanced AT2-like phenotype or mature type 2 cell phenotype, expressing more surfactant protein C and ABCA3. Now, you may be wondering how this ties into IPF, as we did this in the developmental model. Essentially, if we can determine what drives normal type 2 and type 1 cell differentiation and aberrant type 1, type 2 cell differentiation, we can correlate that to disease. We know that YAP is active during repair, and we know that YAP activation increases expression of AT1 and proximal airway epithelial cell markers now. So what is the role in IPF? And this leads us to our future directions, which may bring in some resemblance to what we've shown earlier, in that in the normal donor lung, again, there's normal type 2 and type 1 cells that are distinct, whereas in IPF, there's a presence of these HOPX, ABCA3 dual positive type 1, type 2 cells, similar to what we see in our YAP active mouse, in which we have HOPX positive and surfactant protein C positive cells showing incomplete differentiation, and the presence of adrin aquaporin 5 RNAs being upregulated in these YAP active mice, as well as proximal airway epithelial cell markers such as CGB3A1, 1A1, and MUC5B. So this brings us to the working model, in which we know that as YAP activates type 1 and type 2 cell markers and increases airway epithelial cell markers, this ties into the abnormal epithelial cell repair that induces and believed to drive interstitial lung diseases such as IPF. Future work will use these mouse models I've demonstrated herein, in which we can either activate or delete YAP and type 2 cells, in conjunction with the well-established bleomycin lung injury model to determine the role of YAP in fibrotic lung repair. And with that, I'd like to thank my various mentors, both currently John Kropsky and Tim Blackwell at Vanderbilt, as well as during my postdoctoral work under Jeff Whitsett, the great group that's built around them, as well as collaborators and our funding sources. Thank you.
Hello, my name is Luis Rodriguez. I am a postdoc in the lab of Dr. Michael Beers. I would like to start by thanking the PFF for inviting me here today and the moderators for chairing this session. I appreciate the unique nature of the PFF Summit, and I will do my best to communicate our findings to the technical and non-technical audiences in the room. Today, I will be discussing intervention studies in our novel mouse model of IPF. Our lab studies the role of the alveolar type 2 cell, the AT2, in pulmonary disease, with a current focus on IPF. The role of the epithelial cell in IPF has been a rich source of debate and discussion since the characterization of this disease. As this group knows, there are two FDA-approved medications for IPF, nintedanib and pirfenidone. Both compounds are referred to as antifibrotics due to their observed impact on the fibroblast. This image here is from a recent review published by the lab, which illustrates the role of epithelial dysfunction in disease development and progression. As you can clearly see, the primary target of these antifibrotic therapies, fibrogenesis, is depicted at the bottom of the image. The location is indicative of the terminal nature of this process. That is to say that fibrosis is the end of a complex disease process. There is a rising consensus in the community that intrinsic dysfunction in the AT2 cell is an underlying initiator of disease, either as a susceptibility factor or direct recruiter of injury agents, including overactive immune cells. Thus, the attention of some in the community has steadily turned to understanding the critical role of the epithelium in IPF. With that in mind, a major contribution to the field from the Beers lab has been in the characterization of clinically relevant mutations in the surfactant protein C gene, a protein made exclusively by the epithelium of the lung. Of particular relevance to this talk is the I73T point mutation, which is associated with IPF. Cell lines expressing the mutant isoform mistraffic and accumulate SPC at the plasma membrane. Cellular attempts to degrade this protein through macroautophagy fail, with additional accumulation in the late endosomal compartment. Finally, this inhibits autophagy in the cell, promoting the kinds of intrinsic stress and aberrant cell quality control that have been well documented in IPF. Capitalizing on this biology, the lab developed a mouse model of IPF using conditional expression of the I73T isoform. In short, we have a mouse that makes very little SPC due to a neocassette found in exon 1 of the SPC locus. However, upon administration of tamoxifen, the neocassette is excised from the locus, and expression of the mutant protein quickly ensues. Within 3 to 7 days post-tamoxifen induction, we observe significant immune cell infiltration, causing lung injury, which is followed by fibrotic remodeling beginning at around day 14. Within 4 to 6 weeks, we observed significant fibroblast expansion and collagen deposition, reminiscent of human disease. This model of fibrosis was first published in 2018 and stands in stark contrast to the bleomycin models as an example of the fibrotic process driven by intrinsic epithelial stress rather than some extrinsic agent such as bleomycin. The shortcomings of the bleomycin model have long been known, and given the mounting evidence for disruption to epithelial homeostasis as an underlying driver of disease, multiple groups have turned to using models with intrinsic epithelial stress. In this environment, and with this model now available to us, our group asked a very important and straightforward question. Does the current therapeutic standard in the clinic work in our model? I want to be as clear in what we are asking as possible. As a community, we know the value of antifibrotic therapy to the patients battling this disease every day. And while these therapies don't represent a cure, they are our best tools in the fight against disease progression. Thus, if I'm suggesting that we have a more accurate model of the disease process, then we had better make sure that the gold standard in therapeutics has an effect in our model before we start using this platform to test the next phase of novel molecules. Now, we didn't simply want to test nintedanib. We also wanted to find a molecule that has been proven to dramatically reduce lung fibrosis in mice. For that, we took the suggestion from our industry partners at Bristol Myers Squibb on the compound SB525334. This molecule is a highly selective antagonist of the ALK5 receptor, also known as the TGF-beta receptor 1. It has been demonstrated in multiple single-hit models of disease to effectively rescue the mouse pulmonary phenotype. And really, TGF-beta signaling has always been a major point of interest in the field. In fact, the mechanism of action of pirfenidone seems to be mediated by TGF-beta signaling. Unfortunately, while direct TGF-beta inhibition works quite well in mouse models, a highly specific TGF-beta antagonist has not made it into the clinic, primarily due to an equally important role for TGF-beta signaling in inflammation. You can see the general mechanism of action for both compounds here. And there is a bit of overlap between the two in non-canonical TGF-beta signaling through RAS. I'll come back to this overlap in the end, but I'll preview our findings by suggesting that TGF-beta is so integral to the disease process that part of the efficacy of nintedanib must stem from the overlap between these two compounds. Okay, so how did we perform our study? We really wanted to perform an intervention that would model what happens in humans. That is, a person comes to the doctor with symptoms and doesn't start taking treatment before they ever get sick. So to do that, we induced the mice and allowed them to go through the majority of the inflammatory injury before starting them on either once-a-day nintedanib or twice-a-day SB5334. A quick aside, I will start referring to TGF-beta inhibition rather than the numerical designation of the compound from here on out for the sake of simplicity. After initiation of therapy, we follow the mice over the remaining 16 days of the model, and we collect daily weights. Our evaluation of the disease process is done through the 4 endpoints depicted here. I'll briefly summarize now, but discuss more in detail in the ensuing slides. First, a general evaluation of health and mortality, which includes daily weight loss. A measure of lung function at the end of the model using FlexEvent. A collection of BAL fluid to measure markers of inflammation and lung injury. And finally, markers of lung remodeling and fibrosis, including collagen deposition and expressing of extracellular matrix genes. As we watched the mice going through these studies, we recorded daily weights as a surrogate for the progression of disease. While weight loss is not a readout for a specific molecular mechanism in our model, generally a mouse that is doing worse is going to lose more weight. Looking at the entire weight curve for an nintedanib intervention, you don't see a dramatic difference in the weight curves. However, the mice taking nintedanib did lose less weight, and the physical appearance of the mice in the intervention arm was improved. In contrast, the TGF-beta intervention has a dramatic and significant effect on the weight loss almost immediately after the intervention was started. The difference was very evident to us. As time went on, it was easy to differentiate between the mice undergoing the intervention and those in the vehicle group just by looking at their activity in the cages. Coupled with the differences in weight loss between the two interventions, there was also significant impact to survival. Kaplan-Meier analysis shows a survival advantage to the nintedanib intervention, with an even greater effect with the TGF-beta intervention. While nintedanib does not reach significance in the study, the data is consistent with observations in other models, where the sample size of the study needs to be larger in order to account for the effect size of the compound. The next readout we were able to analyze was right at the time of lung collection. For those in the audience not familiar with the flex event, it is an instrument that is basically able to push air into the lungs of mice under controlled pressure. The idea here is that given a certain amount of resistance in the lung, a given force will allow for a specific volume of air to be pushed into the lung. The greater the resistance in the lung, the less air will be able to enter. Thus, we can get volume changes for a stepwise pressure addition. The resulting metric, static compliance, is graphed here. In general, for the age and size of our mice, we get static compliance values between 0.06 and 0.08. You can see in gray that after 4 weeks of our model, the value drops to between 0.02 and 0.04. As with the previous data, nintedanib intervention shifts the compliance, with some mice returning to the baseline compliance of the control mice, and some mice experiencing only a mild recovery in compliance. However, with the TGF-beta intervention, we see significant rescue of the static compliance as nearly all of the mice in our study fell into 0.06 to 0.08 block. The title of this talk suggests that we are benchmarking interventions in our model, and this certainly reinforces the idea that for the metric of static compliance, the benchmarks are clearly defined. During the lung collection, we are also able to sample the BAL fluid from each of the mice. Here we are looking at 3 different things. First, total number of cells in the BAL as a surrogate for inflammation and immune activity. More cells in the BAL is consistent with inflammatory phenotypes. We also look at the amount of protein that is found in the BAL. This is a surrogate for lung injury. The idea here is that an injury to the lung will limit the barrier capacity of the epithelium and the endothelium, and proteins found in the serum of the mouse will leak into the lung. Finally, we also look at soluble collagen. As part of the fibrotic remodeling process, collagen is secreted into the extracellular space with a certain soluble portion. In data I won't show here, we have validated this by measuring soluble collagen during an inflammation LPS challenge and did not see increases in this metric. So what does this data tell us? Well, first, if you look at lung injury, we see that both interventions decrease total protein in the BAL and to comparable levels. This is a nice piece of data that when paired with static compliance, suggests our mice retain more barrier function with intervention. Total cell counts also show a decrease in total cell number. However, neither intervention reaches statistical significance. Based on our understanding of the mechanisms involved, it makes sense that the antifibrotic therapy and TGF-beta inhibition did not significantly impact our inflammation metric. However, I can tell you the data is not quite that clear. Under TGF-beta intervention, the composition of cells in the BAL changes from lots of injury-causing neutrophils to more macrophages, an interesting observation that reinforces the importance of immunity in both injury and recovery. Finally, we look at the soluble collagen in the BAL. As is becoming a trend in this data, nintedanib mildly reduces this marker, with TGF-beta intervention reaching statistical significance. Rather than suggesting this means that there is less collagen in the lung, let's just take a quick look. So the final piece to our analysis are markers of fibrotic remodeling. In the center of the slide, we have some representative images from histology collected from our mice studies. At the top, we have a stained image of the entire lung. In a healthy lung, we expect to see a lot of empty space, which allows for expansion of the alveolar sacs during normal breathing. A fairly simple way to distinguish between the specimens is just to look at the ratio between purple and white in the picture. Increased purple in the images suggests more cells in the lung, consistent with injury, inflammation, or fibrotic remodeling. As you can see here, the vehicle lung is quite cellular, while there is a moderate reduction in cellularity under nintedanib intervention and a greater reduction with TGF-beta intervention. But we can also measure the amount of fibrillar collagen in the lung using picoserious red, a dye that binds fibrillar collagen and fluoresces red. We quantified multiple sections in all of our mice and show that under both interventions, there is a significant reduction in fibrillar collagen in the lung. To get an idea of what is going on in the lung, we also looked at gene expression in some lung lysates. Specifically, we looked at a gene panel associated with the secretion of extracellular matrix, a key role of the fibroblast during remodeling. I won't belabor the findings, which to this point you probably expect. But again, we see that nintedanib has a moderate effect on most of the genes in the panel and significance in only one, while TGF-beta intervention significantly lowers the expression of all genes in this panel. Overall and in summary, we have three major conclusions. The first is that intervention studies in this model of disease are feasible, and the benchmarks we have chosen recapitulate what has been found in previous models. Second, we demonstrate that the complexity of the disease process requires us to look at multiple endpoints across physiology, inflammation, and fibrotic remodeling to understand the extent of effect that a compound may have on the mouse and potentially the patient. Finally, I think that even though direct TGF-beta antagonism has yet to produce a successful molecule in the clinic, these studies demonstrate the importance of this pathway in disease and stress a need for characterization in any future intervention studies. Finally, I'd like to acknowledge all of the people who worked on this project. Studies like this take a lot of effort and they take a lot of hands. Everyone in the lab was instrumental in these findings, as well as an acknowledgement to our collaborators at Bristol Myers Squibb, who generously provided the TGF-beta inhibitor, as well as expertise on dosing requirements and expectations. Also to our funding agency at the NIH. Thank you very much.
Hello, everyone. This is Yi from Henry Ford Health System. I'm a research instructor at the Department of Dermatology. Today, I will present my work about the loss of microRNAs in alveolar macrophages attenuates bleomycin-induced pulmonary fibrosis. Recent linear tracing studies have shown that most of the adult tissue-rendered macrophages, including the alveolar macrophages, also called TRAMs, are derived from embryonic yolk sac progenitors and fetal liver monocytes. They can self-renew independently of bone marrow hematopoietic stem cells in a steady state. However, under stress or inflammation, bone marrow-derived circulating monocytes can differentiate into alveolar macrophages, also called MOAM. Here showing the role of the alveolar macrophages in the pathogenesis of PF. Upon exposure to stimulation like bleomycin, the activated epithelial cells and the endothelial cells can release cytokines, interleukin-33, which drive the alveolar macrophage polarizing into the M2 alveolar macrophages, which producing high levels of cytokines, chemokines, and growth factors that drive the activation and the differentiation of fibroblasts, leading to the pulmonary fibrosis. On the other hand, the recruited inflammatory cells, including the NKT cells, Tregs, ILCs, can also either promote or inhibit the polarization of the alveolar macrophages, which in turn regulate their recruitment into the fibrotic tissues. Thus, alveolar macrophages play a pro-fibrotic role in the pathogenesis of PF. MicroRNAs are a class of small non-coding RNAs that negatively regulate the gene expression by directly target their genes, by directly targeted mRNAs. The microRNA processing pathways has long been viewed as a linear and universal to all the mammalian microRNAs. A few key proteins are involved in the microRNA processing, including the DROSA, DASR, and AGO2. I'd like to highlight the protein DASR, which cleaves the pre-microRNA hairpins to the mature length. The deletion of DASR can lead to almost a loss of microRNAs in the cells. In this study, we will use the DASR control mouse as one of the working models, which we'll describe later. A few key factors have been reported to regulate alveolar macrophage development and homeostasis, including the MCSF, GM-CSF, TGF-beta, and the PPR gamma. Interestingly, these factors are potentially targeted by several individual microRNAs as shown in this table, suggesting that the microRNAs may be involved in the alveolar macrophage development and function. To determine whether microRNAs are required for alveolar macrophage embryonic development, we generated CS1R DASR conditional mice, in which the microRNAs are deficient in T-resident macrophages and the monocytes at embryonic stage. Using flow cytometry, we found that the FO80 low, CD11B low population defined as alveolar macrophage precursors was significantly reduced in the dicer-knockout mice at E18.5 and P0, suggesting that micrornas are required for the alveolar macrophage embryonic development. However, whether the micrornas regulate alveolar macrophage homeostasis and function remains unknown. Our recent studies showing that the alveolar macrophage have a distinct microRNA expression profile compared to the fetal alveolar macrophage precursors using the microRNA array. We also found that there are dynamic changes of microRNA expression in the TRAMs and the MOMs during the experimental PF. These observations lead to our central hypothesis. Micrornas might play an important role in alveolar macrophage steady-state homeostasis and its function during the experimental PF. To test this, we generated C11C Cray dicer conventional knockout mice, in which the micrornas are deficient in alveolar macrophage after birth, given that C11C only highly expressed in alveolar macrophage postnatally. We will test both naive and bleomycin-induced PF models using these mice. In the naive mice, we found that both frequency and absolute number of the alveolar macrophage are dramatically reduced in the C11C dicer-knockout mice compared to wild type, along with the increased expression of C11B, which is downregulated in mature alveolar macrophage, suggesting that the mature alveolar macrophage steady state maintenance largely depends on the microRNAs. Additionally, we also look at the proliferation capacity of alveolar macrophage by the KSCT7 staining, which showing a comparable frequency between the wild type and the dicer-knockout mice. We also look at the apoptosis by using the annexin 5 and the 7AD staining of the alveolar macrophage. We found there is an enhanced early and late apoptotic cells in the dicer-knockout mice. This result suggesting that the micrornas may control the alveolar macrophage turnover by regulation of cell apoptosis instead of the proliferation. To determine whether micrornas are required for the alveolar macrophage regeneration from bone marrow, we established a bone marrow chimerical mice. Briefly, we co-transfer the donor cells from the wild type or dicer-knockout mice and the competitor's bone marrow cells from the SGL mice into the irradiated SGL, B6 SGL recipient mice. 12 weeks later, we analyzed the compensation of the alveolar macrophage in the bone marrow chimerical mice. We found that the alveolar macrophage are radiosensitive, and they can be regenerated from the wild type, but not dicer-knockout bone marrows, suggesting that the micrornas are required for the alveolar macrophage regeneration from the bone marrow under stress. To determine the role of micrornas in alveolar macrophage function during the experimental PF, we evaluated the fibrogenesis after administration of the bleomycin into the both wild type and CD11C dicer-knockout mice. We found a severe body weight loss in the wild type mice after the bleomycin treatment, along with the increased long hydroperoxylene content and the TGF-beta production in the lavage in the wild type mice. However, these pathological changes was dramatically reduced in the dicer-knockout mice, suggesting that the loss of the microRNA in alveolar macrophages prevents the bleomycin-induced PF. Similarly, we also observe less cell infiltration and less collagen contact in the lungs of the C11C dicer-knockout mice compared to the wild type, further supporting that the loss of the microRNAs in alveolar macrophages prevents the bleomycin-induced PF. Consistent with the previous studies, we can also distinguish the TRAM and the MOM in the fibrotic lungs by their differential expression of the select F and the CEMB surface expression. Interestingly, we found that the number of the TRAMs were dramatically reduced in the destinocta mice, but the number of the MOMs remains unchanged between the wild type and destinocta mice, suggesting that the micrornas are required for the TRAM homeostasis, but dispensable for the MOM turnover during the bleomycin-induced PF. We further look at the cell preservation hypothesis, and we found the increased apoptotic cells in the TRAMs, but not for the MOMs. Also, the preservation capacity is comparable between the wild type and knockout for both TRAMs and MOMs. To further determine the mechanism by which the microRNA regulate the AM function during PF, we isolate the T-cell resident alveolar macrophage from the naive mice for both wild type and destinocta mice, and also isolate the TRAM and the MOM, and from both wild type and destinocta mice after the bleomycin injury. RNA sequencing analysis showed identified two clusters that were either upregulated or downregulated and during the PF, regardless of the mouse genotype. The genome analysis further identified the shared enrichment in gene sets associated with the cell cycle, metabolic process, the RNA splicing, and the protein phosphorylation in both TRAMs and MOMs in mice given the bleomycin compared to the naive mice. Interestingly, when we compared the transcriptional levels between the wild type and the knockout mice in the TRAMs and the MOMs, we found hundreds of genes were dysregulated and uniquely expressed by either TRAMs or MOMs, suggesting that the microRNA deficiency leads to the divergent transcriptional changes in the TRAM and the MOMs during the PF. IPA analysis further identified the multiple canonical pathways that were differentially impacted by the microRNA deficiency in TRAMs and the MOMs. We did find several activated pathways in the TRAMs as well as inactivated pathways in the MOMs. In summary, microRNAs are not only required for the embryonic development of alveolar macrophage, but also required for alveolar macrophage maturation, maintenance in a steady state, and regeneration from the bone marrow under stress. Importantly, microRNA deficiency inhibits the bleomycin-induced fibrogenesis, likely through inhibition the turnover of the TRAMs and regulation of the different signaling pathways in both TRAM and the MOMs. In conclusion, the microRNAs are the key epigenetic mediators that differentially regulate TRAM and MOM maintenance and function in the pathogenesis of PF. Studies are in progress to characterize the role of individual RNAs in AM development and function in PF. Our recent studies identified 4 distinct alveolar macrophage subset in the steady state using the single-cell RNA-seq and the single-cell ATAC-seq. Thus, it would be interesting to see if the microRNAs play a subset-specific role in alveolar macrophage function during the PF. In the end, I'd like to thank to my supervisor, Dr. Qingsheng Mi, for his great support in this project. Also thank all the lab members and the collaborators. Thank you for your attention.
Hello, and thank you for coming to today's session. I am Maggie Freeberg. I'm a postdoc in the lab of Dr. Patricia Syme at Virginia Commonwealth University, and I'm excited today to present to you on our study involving mechanosensing by piezo2 is associated with metabolic reprogramming in human lung fibroblasts. Thank you to the organizers for giving us this opportunity to present our work, and we have nothing to disclose at this time. Idiopathic pulmonary fibrosis is a devastating disease with no true cure and a prognosis worse than most lung cancers. IPF destroys the normal lung architecture, resulting in impaired function and difficulty breathing. There are 2 FDA-approved drugs that slow the progression. However, there's still a shortfall of current therapeutic options and an unmet clinical need for additional therapies that halt fibrotic progression. IPF is a multifactorial problem with several compounding feed-forward loops, and we believe that targeting a single pathway or loop alone will be insufficient to truly halt progression. My research focuses on understanding what drives progression of fibrosis, and today I'm going to tell you about 2 pathways we think might be great targets for future drug development. One of the reasons that pulmonary fibrosis is difficult to treat is targeting a single driver is not always successful. I'm going to touch on how fibrosis affects cellular metabolism and promotes fibrosis via an increased lactate in an unexpected way, as well as how mechanical forces promote fibrosis. But what I'm really interested in is how these 2 pathways interact and influence one another to drive fibrosis and the potential to identify novel therapeutic targets. Previous work in our lab and by others has unexpectedly demonstrated abnormal cellular metabolism as a contributor to fibrosis. Later in this talk, I'm going to link this altered metabolism to changes in stiffness. Abnormal lactate metabolism was first identified about a decade ago as being elevated in the exhaled breath of IPF patients. And this was a surprising finding as we don't normally think about lactate accumulation in the lung. This study went on to demonstrate that lactate turns normal fibroblasts into scar-forming fibroblasts called myofibroblasts. And this is through a process of pH-dependent TGF-beta activation. TGF-beta, as you might recall, is a master regulator of fibrosis. And this creates a feed-forward loop of enhanced myofibroblast differentiation, lactate production, and TGF-beta activation contributing to progressive fibrosis. The next step in the translational pipeline is to block these pathways that have been identified and see if we can prevent fibrosis in preclinical animal models. Here's a piece of data from our lab demonstrating that inhibition of lactate is an effective therapy to reduce pulmonary fibrosis. In this commonly used model where bleomycin induces fibrosis, we see that in the vehicle bleomycin group, there's increased extracellular matrix with the blue staining and accumulation of cells compared to the normal lung. When lactate's inhibited, there's a substantial reduction in the development of fibrosis. Tissue mechanics are also important because cells behave differently on different tissue stiffnesses. Stiffness in the fibrotic lung will increase due to increased quantity of the extracellular matrix as well as changes in the ECM composition. Normal lung is very compliant and soft with stiffnesses between 0.2 and 5 kilopascals. And then at the other end of the spectrum, we have bone. We also have plastic and glass, which is our standard tissue culture conditions. In the fibrotic lung, the stiffness will increase to 10, 20, or even 30 kilopascals in early fibrosis. However, in the dense fibroblastic foci, stiffnesses upwards of 60 to 100 kilopascal have been observed. Other groups have published that these increases in stiffness affect myofibroblast differentiation, cell migration, proliferation rates, and matrix production. The objective of this work is to investigate the effect of matrix stiffness on metabolism in human lung fibroblasts, and we hypothesize that the increases in stiffness will drive profibrotic metabolic change. And we accomplish this using an in vitro model system of primary human lung fibroblasts that have been isolated from non-fibrotic donor biopsies. We're using silicone substrates that mimic physiologic stiffnesses, utilizing 2 kilopascals as our healthy lung range, compared to 32 and 64 kilopascals, which will be used as our fibrotic stiffnesses, and then compared to tissue culture plastic, which is super physiologic and very stiff with multiple orders of magnitude increases in stiffness compared to normal healthy lung. Our outcome measures will look at metabolic derangements and will assess gene and protein expression, measure lactate in the media, as well as perform untargeted metabolomics analysis. Glycolytic metabolism and oxidative phosphorylation are processes for cells to generate energy from glucose. When oxygen is present, the primary pathway is through the TCA cycle and oxidative phosphorylation. When there's no oxygen present, there's this alternative pathway called glycolysis that results in less energy and produces lactic acid as a byproduct. Unusually, some cells will go through this alternative glycolysis pathway even when oxygen is present, and this metabolic change is one of the features of fibrotic fibroblasts. These Western blots are showing proteins associated with fibrotic metabolic changes, and here I'm looking at pyruvate dehydrogenase kinase because they block the normal TCA cycle. I'm also looking at lactate dehydrogenase A because it's an enzyme that helps produce lactate, as well as assessing monocarboxylate transporters that facilitate the export of the lactate from the cytosol into the extracellular space. These Western blots demonstrate an upregulation of these components as substrate stiffness increases. We next ran untargeted metabolomics to determine the full metabolite profiles from cells cultured on different stiffnesses. Here's a heat map of our results. The rows indicate the unique metabolites. The columns represent the fibroblasts cultured on individual stiffnesses from unique human donors. The overall expression level is indicated by the color bar, where blue is low expression and red is high expression. The rows are then clustered based on similarity of expression patterns across the substrates and donors. In our bottom group here, we identify metabolites that are changing between tissue culture plastic and the silicone substrates, but do not change much between 2 and 64 kilopascals. In our middle group here, we identify metabolites that are decreasing with increasing stiffness, but can also distinguish between fibrotic and non-fibrotic stiffnesses of 2 and 64 kilopascals. Our top group identifies metabolites that are increasing as stiffness increases, but are further enhanced by the addition of TGF-beta to the culture system. To understand how these observed metabolite changes are impacting key biological processes, we ran pathway enrichment. And in this figure, the pathway is listed on the left, and the green dot indicates an enrichment score, with the farther to the right and the larger the green dot, the more significant the enrichment. And we observe significant enrichment in pathways related to amino acid metabolism, carbohydrate metabolism, and lipid metabolism, as well as a few others. Two pathways I want to draw your attention to are the R gene and proline metabolism, which is significantly enriched as we increase stiffness. And this is expected because as we increase stiffness, we're going to enhance myofibroblast differentiation, which will increase extracellular matrix production by these cells. And proline metabolism is critical for collagen synthesis. Additionally, in carbohydrate metabolism, we're observing a significant enrichment of glycolysis and gluconeogenesis, as well as pyruvate metabolism, indicative of the glycolytic changes that we've been observing. In addition to validating our hypotheses, it will be exciting to explore and uncover these pathways that are not predicted and identify new targets, connecting these alterations in stiffness with metabolic reprogramming. Given our findings that stiffness regulates cell metabolism, we're interested in how these cells sense their mechanical environment through a process called mechanotransduction. In this process, receptors on the surface of your cell will sense changes in physical forces, converting those forces into gene expression changes, and in this case, contributing to the progression of fibrosis. Given that we're seeing increases in excess lactic acid, we're interested in the mechanosensitive ion channels that can sense these changes in force, but also be specifically activated by pH. We're specifically interested in looking at the receptors piezo1, piezo2, and TRPV4. There are other mechanosensors that are important, such as integrins and G protein coupled receptors, but they will not be our focus. To determine if these mechanosensors of interest play a role in metabolic change, I used siRNA to inhibit gene expression and measured the lactate in the media. When piezo2 is blocked, I observe a 25 to 30% reduction in lactate on all stiffnesses. And this indicates that blocking the mechanosensor piezo2 reduces the cell's ability to sense their substrate, blunting their lactate response. I've also looked at piezo1, TRPV4, and TRPV4, but they appear to be less important. We also ran a microarray of 24 different genes associated with fibrosis and metabolism and mechanosensors. In this heat map, the expression levels are still indicated by the color scale, where red indicates high expression and blue indicates low. In this case, rows represent our genes of interest, where columns represent the time that the cells have been cultured on these various stiffnesses. And then they're clustered according to similar patterns in gene expression. The pattern in the top half of the heat map are 3 clusters of genes that start out low and increase over time, but are somewhat delayed on the silicone substrates compared to tissue culture plastic. And this is comparable to the pattern that we observe in this line graph for piezo2 expression over time. Our top cluster here contains the genes PDK4, FOXO1, and piezo2. Piezo2 is that mechanosensor of interest and indicates that they're potentially regulated together. Recall that PDKs and FOXO1 are negative regulators of the TCA cycle. Our next cluster contains the other mechanosensor of interest, TRPV4, and some fibrosis-related genes. Our bottom cluster are a number of genes that do not have as much of a pattern to them. And overall, we use this microarray approach to identify which genes may be regulated together and have interesting changes associated with their alterations in substrate stiffness. We're very excited to share these new concepts that may be contributing to fibrosis and these interactions between mechanical changes and metabolic reprogramming. We think these might be great targets for therapy in the future. We've identified that increased stiffness leads to metabolic changes, driving the production of lactic acid. And we've implicated the mechanosensor piezo2 as being involved with linking stiffness and metabolic alterations. Ongoing work is looking at more nuanced activation and inhibition studies of these mechanosensors, as well as targeting the therapeutic efficacy of inhibiting these in preclinical animal models of fibrosis. Thank you for your time and attention today. I want to take a moment to thank our funding sources, our incredible research team at BCU, and of course, the patient support groups that we've been affiliated with and the patients who have donated the samples making this research possible. Thank you.
All right, thank you so much for the opportunity to talk with you today. My name is Bhavika Kaul, and I am a new assistant professor in the Division of Pulmonary and Critical Care Medicine at the University of California, San Francisco. Today, I will be sharing my work examining the association between Agent Orange exposure and idiopathic pulmonary fibrosis, or IPF, among US veterans. I have no relevant disclosures. And what I would like to do is start by reviewing the pathogenesis of IPF with a particular focus on our current understanding of the role of ambient exposures. Then we will review our research examining whether military exposures, specifically Agent Orange, could be a risk factor for IPF among the US veteran population. And we'll end by reviewing the policy implications of this work and future directions. As many of you know, IPF is a progressive fibrotic lung disease associated with high morbidity and mortality. And in our current conceptual model of IPF, pulmonary fibroproliferation occurs in genetically susceptible hosts with age as the lung is exposed to cumulative stress. This can be intrinsic exposures such as acid reflux or alterations in the lung microbiome, or it can be extrinsic exposures such as smoking, air pollution, or workplace exposures. Of these, smoking is probably the best defined risk factor, with observational studies suggesting a greater than twofold increase in risk among tobacco users. However, there is a paucity of data about the role that military exposures may play in the development of IPF. And that's what we'll focus on today. Our understanding of the role that environmental and occupational exposures play in IPF is growing. Pollutants are hypothesized to trigger alterations in mucosal surfaces, induce oxidative stress, and modify epigenetics, which may contribute to the pathogenesis of IPF. And recognizing this growing body of literature, the American Thoracic Society and European Respiratory Society published a joint statement in 2019 that specifically examined the impact of exposures on the burden of non-malignant respiratory disease. In their analysis, specifically looking at IPF, they found that the pooled population attributable fraction of workplace exposures on IPF was 26% for vapors, gases, dusts, and fumes, with variability across subtypes. Now, building on this literature, our group was interested in evaluating whether military exposures were also associated with a higher risk of IPF. Of the military exposures, Agent Orange in particular has gained attention due to a combination of toxicology data that has documented its harmful effects and complaints by veterans about their own health. Agent Orange was a chemical defoliant named because it was stored in barrels identified by these orange bands during the Vietnam War. It contained the unintended contaminant dioxin, and the association between dioxin and malignancies such as soft tissue sarcomas, lymphomas, multiple myeloma has previously been described, but there is limited data on dioxin and respiratory disease. So in this study, we sought to evaluate whether exposure to Agent Orange was associated with an increased risk of IPF among US veterans who served in Vietnam. And for this study, we leveraged the strength of the VA's electronic health record system. The Veterans Affairs healthcare system is the largest integrated healthcare system in the United States. It's comprised of over 130 hospitals and more than 1,000 outpatient clinics. And together, this network serves over 9 million veterans each year. All data from these clinical encounters is extracted, transformed, and loaded into a central data repository called the Corporate Data Warehouse, which can subsequently be utilized by researchers to answer clinical questions. And so for our work, we used this data repository to identify a cohort of patients who served in Vietnam. We started with 10.7 million unique veterans enrolled in the VA between 2010 and 2019. From this cohort, we restricted the cohort to the 3.6 million male veterans who served in Vietnam. Among these, we identified approximately 71,000 that had an ICD diagnosis code for IPF and no subsequent codes for an alternative interstitial lung disease. And then the remaining 3.5 million veterans did not have an IPF diagnosis code during our time period. We then went back to the corporate data warehouse to identify veterans who had an Agent Orange flag. The flag is derived from military discharge paperwork, and it signifies that the patients were boots on the ground in Vietnam. Because of the widespread spraying and unpredictability of wind dispersal patterns, it's not possible to quantify the precise degree of exposure for an individual veteran. And then, as I previously described, we used an electronic health record-based phenotype to identify our primary outcome of interest, which was IPF. We also extracted clinical covariates, including age, race, ethnicity, rural residence, smoking history, and military service line. And we extracted these data points because we wanted to be able to control for known IPF risk factors. And so what we found was that about 26% of the 3.6 million veterans who served in Vietnam had presumptive Agent Orange exposure based on this Agent Orange flag. And now examining the differences between the two groups, what we found was that the mean age was pretty comparable between those who had no Agent Orange exposure and those veterans that did have presumed Agent Orange exposure. However, patients with Agent Orange exposure were more likely to be white, non-Hispanic or Latino, and live in rural areas. Tobacco use was also comparable between the Agent Orange and no Agent Orange exposure groups. And then what you'll notice is, not surprisingly, when we look at military service line, the Agent Orange group was predominantly composed of Army and Marines because they were the group most likely to have the most significant amount of time with boots on the ground in Vietnam. We found that IPF occurred in 2.2% of veterans exposed to Agent Orange versus 1.9% of veterans without Agent Orange exposure. And this correlated with a 14% increased risk of IPF among veterans with Agent Orange exposure. We then recalculated the relative risk after adjusting for known IPF risk factors, including age, race, ethnicity, smoking history, and rural residence, and found that even after adjusting for these risk factors, there was an 8% increase in relative risk of IPF. We then ran 3 additional sensitivity analyses. In the first, we restricted the cohort to Vietnam veterans who served in the Army as a surrogate for the degree of Agent Orange exposure, since they spent the most amount of time with boots on the ground. We found a slightly higher relative risk of IPF in this cohort. And then in the second sensitivity analysis, we restricted the cohort to those patients who had the most specific diagnostic codes for IPF. And we found similar results here. Lastly, we restricted the cohort to both Army veterans and those patients with the most specific IPF ICD codes, and found a 23% increase in the unadjusted relative risk of IPF among veterans exposed to Agent Orange, and a 16% increase in adjusted relative risk after accounting for these other IPF risk factors. Finally, we also utilized fine-grained competing risk models to examine the time to IPF onset using age as the time scalar and accounting for the competing risk of death. After accounting for this competing risk of death, veterans with Agent Orange exposure were still more likely to develop IPF. Here you see the difference in cumulative incidence of IPF diagnosis over the 10-year study period between those with and without Agent Orange exposure. So what does this mean for us as an ILD community and for the VA healthcare system at large? Well, as I mentioned, Agent Orange in particular has gained attention within the VA over the last few decades. And in response, Congress passed the Agent Orange Act of 1991, which directs the National Academy of Sciences to review new scientific evidence describing an association between dioxin and Agent Orange and health outcomes about every 2 years. This committee categorizes health outcomes related to Agent Orange exposure into 4 broad categories of evidence based on the strength of the data that's available. We hope that our work will add to this literature, and with further study may even support a change in the characterization of IPF. Now, our work does have a number of limitations. First, this is an ecological study and relies on EHR data, specifically ICD codes, to identify IPF, and these have not been individually case validated. However, the conclusions remained consistent with several sensitivity analyses, and in fact were numerically greater when we used more specific ICD codes for IPF. In addition, we used the Agent Orange flag to indicate history of exposure. And as I previously mentioned, due to widespread spraying and unpredictability of wind dispersal pattern, it's not possible to quantify the precise degree of exposure for an individual veteran. Lastly, because this is observational data, we cannot completely rule out confounding. And while these limitations do likely introduce some false positives into our case definition and exposure determination, we expect that such classification error would bias our results towards the null. So the fact that an association is still found is notable. So in conclusion, we found that Agent Orange was associated with an 8 to 16% increase in the relative risk of IPF among veterans as we went from more sensitive to more specific definitions of the exposure and outcome. This work contributes to a growing body of literature implicating the interplay between genetics, aging, and exposure in IPF pathogenesis. Future research will need to validate this association and investigate the potential biological mechanisms that may be involved. Now, this work could not be possible without a multidisciplinary team, and I particularly want to thank Dr. Mary Hooley, who is my VA research mentor, and Drs. Joyce Lee and Hal Collard, who are my ILD content mentors. And on that note, I will pause and I'm happy to take any questions. Thank you.
Well, hello, everyone, and welcome to the question and answer session, which is live following those great poster presentations. So I'm pleased to be able to moderate. I just want to remind everyone in the audience that you're welcome to put questions in the chat, and I'll try to give them to our speakers. And so I'm just going to start now with the first question for Dr. Gokey, which is really thinking about YAP activation or deletion and the driving differentiation of AT1 and AT2 cells. I wonder if you could just maybe address the discrepancy between the organoid data, which seemed fairly clear, compared to the RNA-seq and the ATAC-seq data that was a little bit more of an incomplete differentiation phenotype. What do you think about that?
So functionally speaking, while those organoids, especially the YAP active in particular, do make type 1 markers, HOPX and AGER, indicating a drive towards the type 1 cells, you'll note that they actually have a cuboidal shape. They're certainly not stretched out, anything resembling a normal type 1. So I do believe that they're also not a completely differentiated type 1. I'm working on trying to validate that currently. So basically, I think you need YAP to drive type 1 cell differentiation, but the sustained YAP still leads to abnormal ectopic expression of other airway markers indicating incomplete differentiation.
Thanks, makes sense. I think the next question is for Dr. Syme, who's stepping in for Dr. Freeberg, who I understand had a baby today, so that's a good excuse to miss. And so the question here is, why do you think the cellular metabolic profile is changing due to the changes in the stiffness when you set up your culture model, the cells on different kinds of stiffness, and you get these changes in metabolism? What do you think is really driving that?
Yeah, thanks. Great question. So, that's some of the really good questions we're trying to sort out. We think what's happening is that, stiffness is being sensed by some of these stiffness sensitive, stretch sensitive channels, and maybe in addition to some pH sensitive channels. And that's probably in some ways, signaling to the nucleus through intracellular signaling pathways to drive metabolic change. So, we've got some very preliminary data looking at which of the receptors on the cell surface might be sensing that. And that was why we showed some data on the piezo channels. Others have looked at TRPV4 as a way to sense the extracellular stiffness and drive to myofibroblast differentiation. We're looking at that, but in addition to metabolic change. So we're sort of excited that these pathways might intersect, and actually the changes to myofibroblast induced by stretch may further feed into metabolic dysregulation. So we're very interested in these kind of feed-forward loops and combining the ideas of stiffness driving myofibroblast, stiffness driving metabolic change, and how those two might intersect.
Yeah, I agree. I mean, I think it's so interesting. We really don't know much about why this disease is progressive. And I think the idea of the stiffness driving it is one of the most interesting concepts in that regard. So the next question is for Dr. Yao, and is really, do the microRNAs alter the recruitment of LY6E high monocytes to the lung? This population has been believed to be pro-fibrogenic and somewhat long-lived. And if so, how would that affect the conclusions? Because I think I was struck by the fact that in your presentation, you didn't really see a difference in your MOA-AM population, and you really think it's an alveolar macrophage phenotype. And yet I know some of the work from Sasha Mishra and all has really suggested it's more these inflammatory monocytes. So what do you think about the microRNA regulation of those different populations?
Yeah, it's a great question. Actually, in our CD11C diazonocola mice, and in the naive mice without induced any inflammation or fibrosis, and we did see a dramatic reduction for the resident alveolar macrophage, but not for the other, like monocytes or neutrophils, for other cell types in the non-colonized mice at a steady state. And however, if we induce the fibrosis, we do see a dramatic reduction and for the monocytes, neutrophils. That makes sense because we see a less inflammation, right, in the fibrotic tissues. So since we know that the CD11C clay is only target the resident of the macrophage and the monocyte derived of the macrophage in the lung tissue, but not targeting the monocytes. So our result still the supportive for the function for the the alveolar macrophage and during the the PF. And it will be interesting to look at the monocyte function and in our model too.
Yeah, I mean, I think it's still such an open question, for monocytes. I've been in the field long enough to see myeloid cells, come into favor, go out of favor, come back into favor a little bit. So, but yeah, the idea about these different populations, and of course the markers in the mice and the humans are so different, it's very hard sometimes to know which populations. But I think it's interesting because your work clearly shows alveolar macrophages are important, and I think we've also got other work that shows these inflammatory cells are important, and so that'll be fun to try to figure out and sort out over the coming time.
Yes.
The next question is for Dr. Kaul, and so thank you so much for your presentation on a really important clinical project. And so I guess maybe an obvious question, when you talk about Agent Orange, I think about dioxin and maybe about aryl hydrocarbon receptor signaling as being what might be relevant. But I'm just wondering, what are your thoughts about how Agent Orange might have been sensitizing the veterans to the development of IPF?
That's a great question. And you're absolutely right. That is one mechanism we've thought of. The short answer is that there still needs to be a lot more work looking at the role of dioxin in causing potentially pulmonary fibrosis. There are some animal model studies specifically looking at the aryl hydrocarbon receptor, suggesting that dioxin binds to and activates it. And it may be an important modulator for adaptive immune response in these patients, which may contribute to pulmonary fibrosis through the chronic effects of lung inflammation. But again, that's very preliminary. That's mostly in animal models. And because this hasn't really been looked at in a clinical context until now, our hope is that this will trigger more investigation in this specific area.
Yeah, I know for me, especially, it made me wonder about, alterations of maybe infection in those patients and wondering if the Agent Orange had any impact on maybe, infections, viral infections or things like that might have also been, also driving some of that pathology. So thank you so much.
Of course.
So Dr. Rodriguez, I think my next question is for you. So you gave us a great poster where you talked about an animal model that could be very useful for studying nintedanib and, really any anti-fibrotic effect. And I think we talked a lot at the original poster session about, how important TGF-beta is as a pro-fibrotic mediator. And I think you made the point in your talk just now that TGF-beta signaling is something we should be thinking about as an outcome measure in these clinical trials. So I guess my question to you is, based on the work that you've done and all of the different ways you can look at TGF-beta effects, if we were to try to build into our future clinical trials a measurement to know whether we're impacting TGF-beta signaling, what would you suggest as sort of the right way to do that? I realize that's a huge question, but
Yeah, I mean, that's a great question. I don't know. I think that the problem with TGF-beta is that it's such a broad pathway, and it's really a broad pathway in fibrosis, and it affects so many different cells. And I don't know if we could pick any one particular metric. You could look at the expression, you could look at the amount of active TGF-beta, you could look at maybe circulating TGF-beta. There's lots of different ways to do it. But I think it's the downstream targets of TGF-beta that are the issue. And so I think anything that touches on the downstream targets of TGF-beta is probably a worthwhile pathway and process to look into, maybe a molecule to explore. I'm sorry, that doesn't quite exactly answer your question, but yeah, that's where we go.
Well, I agree. I think it is the pathway we all know is involved. And, I think, you've proved nicely that nintedanib really is a weak TGF-beta antagonist compared to other ones. And that may be why it's useful clinically for sure, because TGF-beta has all kinds of side effects if you block it completely, from the immune standpoint. So, all right, let me go back to Dr. Gokey with my next question. So, what is known regarding the link to alveolar epithelial cell development versus alveolar epithelial cell repair and YAP, and maybe even to add on to that, do you have any idea what's turning on YAP in the fibrotic lung?
Okay, so yeah, we know now between work we've done and work Ed Morrissey's done and Jit Xiao Chen that have all largely been published in the last month, that YAP deletion You need YAP during normal repair processes. It turns on and then it turns back off during both development and repair. One of the main reasons we looked at the time points we did was we're just trying to tease apart the mediators of the YAP signaling to try and alter, because if you activate YAP, bad things happen, as we see in the context of, we know YAP is active and sustained active in IPF, but we also know you can't inhibit YAP because then your type 2 cell will fail to repair normally in the initial regenerative process. So I forgot what the second part of that question was already.
Oh, I think I asked if you had any idea how YAP was actually getting upregulated in the first place. What's the upstream signal?
So that would be, there's two very possible answers. One is, like I said, the chronic epithelial cell injury, and during normal injury repair, YAP turns on. And for some reason, it doesn't turn off. In IPF, the YAP inhibitor is absent, which seems the MST1-2 as upstream is actually missing in a lot of the cells. So therefore, YAP is maintained. The other strong possibility is YAP is activated by mechanotransduction and increased stiffness. So when you get near those fibrotic foci, you might be activating YAP in those epithelial cells, thereby pushing it further through progression.
Okay, thanks so much for that. Let's see, how about to Dr. Yao? Here's a question that asks, so you've shown that if you get rid of dicer, where you impact so many microRNAs, that you have these profound effects. So how are you going to go about trying to figure out which particular microRNAs are really the ones that are driving the phenotype? Do you have any strategies?
Oh, it's a very, very nice question. Actually, we are currently working on that. It's not easy to find the one, individual RNAs and play an important role, right, from that thousands of microRNAs. So actually, we recently did that microRNA array and to look at the microRNA expression profiles, and in the alveolar macrophage, and in the naive mice versus the cells from the PF model. And interestingly, there's several, and the microRNAs is dramatically upregulated. I can give you some clues, and some RNAs like miR-155, and we know that it's playing important role for the macrophage function, right? M1, M2, and also another one is miR-10. And so there's some other ones. I don't want to give all the secrets here, but yeah, definitely we are working on it. Yeah.
Perfect, thanks so much. Dr. Syme, here's a question about thinking about the model system. Does the collagen ECM used to facilitate attachment of the fibroblast contribute to the functional changes? And would you expect the results in your studies to be different if you used a different ECM protein, such as fibronectin? So is it, I guess that's sort of a way of asking, is it stiffness or is it collagen in particular that's important?
Yeah, I think there's a couple of angles to that answer. We haven't actually tested it. As you know, many people use fibronectin and we chose to use collagen. I think it probably does make a difference because depending on the way that the extracellular matrix signals into the cell may have different effects, either in driving proliferation or myofibrous differentiation or metabolism. For example, there are different integrins with different collagens and fibronectin, so it might well be different. We actually haven't tested that, although we know from work from others, for example, that Mitch Oatman's done some very nice work on fibronectin and how that drives differentiation. So I think, and the matrix probably changes over time as well. So although we're initially plating with collagen, the cells, of course, are making matrix. So they're making fibronectin and collagens, and the collagen will change. So it will be cross-linked, the longer it's in collagen, in culture rather. And so that may also drive behavior. So I think the intersections between different kind of matrix, the time it's laid down, and mechanical stress in vivo is going to be really interesting because that changes very dynamically over time. And truthfully, probably different fibrotic diseases have different patterns of matrix expression at different stages. So I think it's going to be really complex to unravel, but very interesting.
Got it. Thank you so much. So Dr. Rodriguez, I wonder if I could go back to you. I think in your presentation, you talked a little bit about the effects of TGF-beta and neutrophils and kind of made the point about innate immune cells being important both in the injury and in the, maybe the progression of the disease. I wondered if you could just maybe touch on that again and remind the audience sort of what your results were with that.
Yeah, that was a really fascinating observation that we made, and certainly a question that we were thinking about in depth here. And so When we treated with the TGF-beta inhibitor and the nintedanib inhibitor, in both cases, we were unable to bring down the total cell counts. However, the makeup of the cell counts was slightly different. So the neutrophils and really the granulocytes in general were decreased in the TGF-beta inhibition arm. And instead of having those granulocytes, instead we had an increase in the number of mononuclear cells, so maybe macrophages. And so it certainly suggests that there's bad players and there's good players in the progression of the disease, and then of course during the remodeling period. And so for our model, we really see that it's really a neutrophil-driven injury. And so there must be some sort of signaling mechanism that, again, involves TGF-beta that promotes chemokines that recruit the neutrophils. And so, if we're trying to explore what that connection is and how that particular aspect of the intervention could be capitalized upon.
Yeah, I think that's going to be interesting because I know there's data about neutrophil nets driving myofibroblast activation, but also neutrophils being important for, clearing apoptotic cells and things like that. So I think that, how the neutrophils are regulated by our drugs that we're testing, as well as in the disease models, I think will be very important. And so, Dr. Kaul, I know we're down to just 2 minutes, but I just want to, I think the VA hospital and the veterans records and all that you showed us, I mean, it's such a wealth of information to be able to look at so many different diseases. I just wonder, kind of what your next questions are that you're going to be pursuing. I mean, obviously the Agent Orange question was interesting, but you mentioned many other environmental exposures. I'm just sort of curious what your next steps are, maybe a future plan question for you.
Yeah, absolutely. So my niche is in health services research. And so it kind of came up as an interesting question as we were doing epidemiological work. And so some of our next questions are actually focused on access to care, access to antifibrotics, quality of care within the VA healthcare system. And then as far as exposures go, just to address that part, there is a burn pit registry, which many people have asked us about whether we are planning on looking at. And what I would say is yes, but not yet, because we know that aging is a significant factor in IPF, and a lot of our burn pit veterans are still on the younger side. So I suspect that that registry will be ready to look at in maybe the next 5 to 10 years upcoming.
Perfect. Thank you. Well, I see there's one last question in the chat. I think it's for Dr. Syme. In terms of stretch mechanisms perpetuating fibrosis, would you advise patients to limit activity to preserve lung function? You have 43 seconds, Patricia.
I'm glad, because it's a dangerous question. I think there's data to show that, keeping people mobile and pulmonary rehabilitation are beneficial for patients. So I think we encourage people to stay active because of all the other comorbidities that happen when you're not active. I think there are very interesting questions about, operating on the lungs of the patients, because you probably really do change dynamics. And if we had more than 16 seconds, I could give you a better answer on that. But I think we do want to keep patients moving.
Well, thank you. I just want to congratulate again our poster presenters for doing a great job. Thank you to the audience for the questions. And there's obviously still a lot of work to do in this disease. So thank you for joining us today. Have a great day.
Thank you.
Thank you.